Welcome everyone to the Emergent Town Hall session for the BIDS PET. Martin is just about to join me over here. We would like to talk to you guys and give you a short introduction to the status of the BIDS PET extension. This has been a work in progress for, I would say, a year and a half, but it has gained a lot of momentum now, and we're very close to actually making a pull request to the main spec. So we wanted to get as much feedback as we can now, and also give you a little introduction to PET BIDS, what are the possibilities that you can do with it, and so on. I have already posted in the chat the up-to-date link to the specification. Then we also had a PET BIDS workshop Monday last week, just before OHBM started, where we had a lot of PET dedicated folk meetup, and we had some presentations about the specification and the validator, also from the maintainers and from the head of the steering group. We had some introduction to BIDS, and then we actually had some carrot, I would call it, presentation, which showcased tools that can be used for data that is put in a BIDS format for PET. So that's something very interesting for the people that want to use it. And the YouTube link to all the videos that we made during this workshop is there. And then we'd also like you to join our discussion on Brain Hack in the Mattermost channel. So I will give my word over to Martin, who will tell you a little bit about the status of the PET BIDS. And please ask us questions as you go. I know there's not so many dedicated PET people here, so please just interrupt and ask, either via the Ask the Question form or just in the chat. We'll try, since we're two people, to post and keep an eye on it. So I'll let Martin take over.
Thank you very much. Good to see you all. Can you all hear me? Yeah. So I just want to give you a brief introduction about the status of the PET BIDS, and I just need to share my screen here. And can you see my screen? Okay, this looks crazy. Let me just try again. I can't. Okay, I have to move it over here 2 seconds. Just to do this. And like this. And then I just have to do this. Okay, because I can't see your way. Okay, now I can see it. Okay, so I just want to go quickly through the status of the PET BIDS, and also to give you that are here in this session like an overview of PET, and I'm gonna make it like short because otherwise I could talk all day about this. So just to get started, I just want to quickly go through like some of the concepts of PET, and also some of the rich sources of data that we have for PET imaging, because that's very important. If you want to make a standard for sharing PET data, you really need to know all the important components of a PET experiment. And this is basically what it looks like. You develop a radio tracer that are specific for a specific receptor in the brain. That could, for example, be the serotonin transporter that we have medication to target. Then you develop the tracer, you inject it into the body, it goes into the brain, and then you scan your subject using a PET scanner. And because this radio tracer is radioactive, it has a decay over time, and that's basically what you're measuring with PET. Now, you're also interested in knowing how the tracer goes from the blood into the brain and gets washed out again. So often you also have a blood component where you measure the activity in blood over time, and that is often referred to as an input function. Now you get your images out of the scanner and they look something like this. And this is purely reflecting the function of the radio tracer or the uptake of the radio tracer. So you also need an MRI to register these two, where you get the anatomical information. And then what you can do is that you can, for each region, you can get the activity over time, often referred to as time activity curves. And then finally, you can use these curves to model the receptor density. And then the final output is something that looks like this. Now, one of the main issues that we have for PET imaging is that they're really expensive. And because they're really expensive, then it's often that low sample sizes are being used. And just to give you an intuition, a common PET scan costs about $3,000 to $5,000. So you don't really have a lot of like money to do that often in a study, and that's why often we have low sample sizes. Now, but it's not because that we don't have the data, because even though a common PET study only have about 20 subjects per group in a study, if you go back, and this is just an example of a radio tracer called DASP, and this tracer targets the serotonin transporter. So you can see that it's been used in a lot of different disorders over time, all from the beginning when it started in 2000, and now up to, this is a chart from 2017. And if you sum over all this, you have up to about 2,700 subjects. And if you look at just the healthy controls, there are about 1,800 healthy controls that are potentially out there that you can use in your analysis. And how do we do that? Well, data sharing is, of course, an obvious thing to obtain this. But the problem is that even though we have the same data, the data is often stored completely different if you're in center A compared to center B. So even though we here in Denmark, we have a PET system, this is a high-resolution scanner, and in the US at Stony Brook, they also have a Siemens scanner, an HR+. The output form is actually the same, but all the information that's related to the PET experiment is often stored in different ways. So for example, this is the way that we store it here. We have it in an Excel sheet. You can see information about the injected dose, other types of information like the specific activity for the tracer. And here it's reported as in that for this, it's 573 megabecquerels. That's the injected dose. And then if you go over here and look how the data was stored here, you see that the injected dose is 16.39. And why is that? That is because it's in a different unit. But if you did not know that, you would have a hard problem with disentangling what kind of information is in this text document. So you have more or less the same information, but often there are deviances between the documents, making it difficult to take this data and use it in an analysis directly. So I hope, I have convinced you that sharing and reusing data are obvious. And this is a slide that I have from Robert Ostenfeld. Some of the nice things about having this specification. First is that it improves data and interpretability and reusability, improves scientific quality and reproducibility, facilitates shared tools and pipeline development, and also reduces stress since you don't have to come up with everything yourself. And this is not new to the PET field, because we started also doing this some years ago, but it's just been taking a lot of time before we have reached at this point where we are now. So we have some conferences each year called the Neuroreceptor Mapping Meeting. BIDS was introduced at this, or at least for data sharing, in Boston in 2016, London 2018, and it should have been in Montreal at this point. And it has also been at the Brain PET meeting in Japan in 2019. Now, what we have established since then is that we have obtained guidelines for what should be in a PET study. And last year, in 2019, we published this study guidelines for the content and format of PET brain data in publications and archived, a consensus paper. And what was really nice about this study is that over 40 researchers from different centers, they joined forces, and they came up with a guideline for what should be in both a paper, but also in a database or in a data format if you want to share PET data. And this is an example of the outcome of the paper. So here you have all the different specifications or labels for each field here that covers both the radioligand development, the timing information, and information about the PET scan. And this is another consensus paper that was also made. It's from 2017, where the radio pharmaceutical people, they went together and wanted to make a common standard for reporting that. Now, the goal for PET BIDS is to, of course, standardize data. So we also have the problem that often this is the output from the scanner. This is the format that we want to put it into. But we need to make sure that what is in the data standard is also reflecting the guidelines that I just referred to previously. And for that, we need to have information about the experiment, the radiochemistry, the timing information, how the data was reconstructed, so from the PET scanner, but also some information related to blood. And I'll just get back to that in a second. So the way that the standard looks like right now is that we have a template for how the data should be structured, and that really reflects some of the major components in the experiment. So first of all, we have a subject label that is also specified in the original BIDS format that gives you like a subject label. That could be subject 01. The next one is that we have the session label. That could be a baseline scan, or for PET, where we often have intervention. So it could either be session baseline or session intervention. Then you also have the task label. So this is if the subject is performing a task inside the scanner. And this is mostly relevant for some of the systems where you have a combined PET-MR scanner, where you want to do some functional paradigm while measuring the PET data. Then when it comes to acquisition, it's not uncommon for PET that we have a dynamic scan. So this is where we measure the uptake over time. So we get different frames. But you could also have, for example, a static scan, like an FDG scan that's common in a clinical setting. And then this would be like a fixed or constant factor. Then you can have it attenuation corrected, or you can have it not attenuation corrected. So this is simply to reflect that you can have different acquisitions during the experiment. Then it's also possible to reconstruct your data using different algorithms. And here we have the rec flag or label. And this is an iterative algorithm, but you could also use filter back projection. So this is simply to reflect that you can have different reconstructions. And then finally, the run label, and I'll get back to that later, or me and Melanie are very interested in discussing this because it's kind of difficult to define, because you can imagine a case where the person has to go to the bathroom during the scan, but because you have made your injection and basically you're measuring the same thing, then if the subject goes back to the scanner and the scan continues, it's actually still the same session, but how should you define it in terms of runs? And we can discuss that later. A final thing is that you also have to keep track of the timing information of the PET scan. And here it also gets a bit complicated, because you have all these different sources of measurements at different time points. So the way that we have defined it is that we have a time 0, and that is defined as, for example, this time point. And we require that all the other measurements have been scaled, or at least, changed according to this time 0. So, for example, it's not uncommon that you often draw the tracer and measure the radioactivity before the scan. Then at a certain time point, you inject the tracer, and this is often the same time point as you start the PET scanner. Then you can have an autosampler that measures the activity in the blood. And often you start this before the PET scan, and it has a high sampling frequency compared to both the PET scan, but also some of these discrete blood samples that you can take. So this is where you take like a manual sample after some time, and you use this information to both measure metabolites and so forth, and then you do a correction on the autosamples. And that is basically what you're using for the final PET data. Now, putting this all together in one piece, this is how it looks like for now, where you capture some information about the PET imaging system. You have information about the injected radioactivity, injected mass, so forth. you have information about the timing, and you also have information about the reconstruction. Then you also have the blood data, which has been specified as a different file, where we have put it into a TSV file with an according JSON file specifying each column in the TSV file. And here you have, for example, the discrete underscore blood TSV file, where you have the time information as the first column, you have the plasma radioactivity on the second, and then you have some metabolite information here. And then you have the corresponding information to the table here in a JSON file. And here you can both define the continuous blood samples from the autosampler, and you can have the discrete values from the discrete measurements. And lots of different data are currently online to look at this. They have different properties, these data sets. So this one, for example, it has the PET data with the tracer SIMBI36. It measures the serotonin 2A availability. Arterial measurements are available for this, both continuous and discrete blood samples. The second one here is for DASP, as I mentioned previously. It also have an MRI, but for this one, there are no arterial measurements available. And then finally, this one where you have also DASP and you have an anatomical T1 weighted, and there are arterial measurements for this one, but not for this one. That's how it is. So finishing up, where to go from here? Well, PET-BIDS is, of course, a community effort, so we need to make sure that the entire community are contributing and helping to get this in the best possible shape. And because often for PET, if we want to make data sharing and also kind of like going back in time, we need to develop local converters that can help each center to get their data into this format, so they're able to share the data with the rest of us. And in order to do that, we have to help them, or at least facilitate data sharing across PET centers, which includes databases, but also help with some of the legal aspects that, especially here in Europe, that we have a lot of potential issues with. And with that, I'm giving the it back to Melanie.
There you go. Thank you, Martin, for a very cool overview over the PET-BIDS. So what Martin has just done is given you an overview of the status. How far are we now? And basically, the issue is now, have we covered all the cases that we want to cover? Have we made this too complicated for people to use? We need to say that we come from a very specified PET lab, and we've mostly worked together with people that do extensive PET modeling, in the sense that this means if you, for example, come into the clinic right now, the most likely case for you to get a PET scan is that you maybe have cancer and then you get a simple FDG scan. This would be the clinical use case. And most of these cases, it's a much simpler modeling, it's much simpler imaging that is being done. So what we would like to know from the community and from the people joining us here is to actually know, what are the use cases that you use PET in? What are the kind of scanners you're working with? What are the tools you're using? So we can make the specification now as broad enough as we would like it to be. And we know that we have a whole spectrum of people, either going all the way from the calculating simple uptake ratios in FDG to doing full kinetic modeling, for example, in the dopamine or serotonin transmitter system. So the use cases are very wide. Also, in terms of what Martin already mentioned, if we look at, for example, the run and session definitions compared to what the original BIDS specification was written for, in PET, we can have a lot more complicated setups because we have this injection of radioactivity, and we can often do injection of other pharmacological challenges. We have quite I would say, a different view of what a run or what a session could mean. And this is something we would like to discuss with you. So we would like to have any feedback right now, if there are comments towards, do you even use PET? What kind of PET modalities are you aware of in your research? We realize that on the OHBM side, most people are probably using it related to Alzheimer's research or maybe aging. So we would like to get some feedback here, and this is why we made this Emergent session. So if you have any comments, shoot now or add it to our document that we link to in the very beginning of the section, or add it on Mattermost. Besides this, we have been also working on actually making a whole pipeline available. Like Martin has said, we have all this very expensive PET data lying around, and now we want to actually get it somewhere. Well, what do we need to push this standard forward? Well, the first thing we need is examples and a validator, and there we're almost on the finish line. So the specification is in the final stages. Martin has already shared with you and given you an overview of some example data that will also be added to the BIDS example list on GitHub. And also we have some data on OpenNeuro that is currently in an older standard of the specification and was uploaded using the BIDS ignore flag extensively, ignoring the whole PET directory, just to be able to share it. But so there's data out there, and we have an implementation, the first one of the validator, and this should be finished soon too. But what we are missing and what we're seeking help on and also input is building a converter. And there I'd like to pull Remi up on stage, if we can do that. If we can pull Remi up. And he can tell you about a project that he has been working on together with Satra in terms of actually constructing the necessary JSON files that could be used, for example, for conversion. And we should also note there was also an OSR talk with respect to BIDSifying any kind of data. This was called BIDS.me and happened last week. And there Nikita also introduced how to use BIDS.me in general. And in our PET workshop, he showed the example on PET. But I'll let Remi take the word right now.
Hey, everyone. Yeah, this was a very sort of last minute thing for me. I sort of met with Melanie last week for on something on the BID starter kit, and I sort of asked her about the guideline, the PET data guidelines that had been released, because anything that has to do with guideline checklists on publishing stuff that's neuroimaging related, it tends to pop up in my Google alerts. So I was like, hey, we need to talk. And I was like, do you by any chance have a table for this? And she was like, yes, I do. Okay, cool. So, and because I was bored this weekend and obviously I was getting way too much sleep, I tried to reuse that table to turn that into like a checklist. So I've been working on something similar for I and some other people, and relying on the work that Sanu and Satra from Repronem have been doing. We've been working on trying to create a checklist app for MRI actually in general, where you just to make sure you report all your methods. And this would just output sort of like a JSON file at the end that would sort of encapsulate all your methods. And the pipeline to create this app is pretty much simple. Like you start from a table, like a CSV, that you turn into a set of JSON LD file. And then you feed that into the app, and it sort of renders this checklist. I will actually post a link because I think we cannot be, I will not be able to share my screen because we're already 4 on stage. But I will actually share 2 links. The first one is the one to the PET version of the app, which is very much work in progress, and we're still fixing things with Satra and Sanu on sort of on the back end, and like there are things that we're still like evolving. And then we met with Martin and Melanie. Was that yesterday? I'm losing track of time. Was yesterday? Okay, cool. To talk about how this could be used and what needs to be tweaked, what needs to be changed at sort of any level. Obviously, what Melanie was telling me is that there are some slight differences already between the table from the paper and the guidelines in terms of units and things. So that's definitely something to that we can definitely make some quick progress on. Actually, let me send you the link to the folder where all those the spreadsheets are, where you can find the one with the PET. So it's just a Google Drive folder, and you have the one for MRI for like the other ones, and the one for PET there. And that's pretty much what I'm working on at the moment. It's just updating this, turn that into a CSV, and then turn that into some like very simple Python script to convert that. And the idea would be just to sort of like, you people can just click through those different items in the checklist, and you can just output a JSON file. The idea, a set of JSON file. The idea now would be how to make that fit into exactly the BIDS type of JSON file we're looking for. But that would be, sort of like one tool to make it easier for people to create those JSON file for. And I think, Melanie, you were mentioning that this could also be very useful just literally on site for people to just keep track and keep a record of what they're doing at every sort of all these moving parts that apparently that the PET specification involved. This is just, yeah. So the idea, like at the moment, like the idea is like getting feedback on the app itself, or how it feels, how it looks, things that might be missing. I know there's some stuff that's just not there. Updating the table so it is now matched with the content of the BIDS specifications that you have. And I think in general, if we could get a couple of people to keep track of this thing on the app front, because I'm more on the MRI front. I know I have some friends who could be in charge of like EEG, MEG. But a bit like for the BIDS community thing in general, where you have people sort of divided by specialty or like by modality, I think it would be good to have someone in there to just keep track of this. So if someone is interested in sort of like giving us a hand on this, would be interested in this definitely. I think, yeah, or if you know anyone who knows some JavaScript, apparently we're also interested in that for the for like just the front end part. So yeah, that's the thing I've been sort of killing time with this weekend, because obviously I had way too much time on my hands. So yeah, that's it for me on my end. I will let you have a look at the app and give us feedback in general.
Thank you, Remi. So this is one of the nice things about the OSR, that we get to meet across modalities, and I do that a little bit. I've been so appreciative of hearing from the original EG and MEG developers, Robert and Guillemar, that I in the steering group see every 3 weeks, and then we discuss, and I often ask them, how did you do this? Because we're at this turning point on how to save things and so on. But it's really great to hear from the other modalities, and that's why we wanted to do the session and ask for feedback. So we the one question that was in there was actually what Remi also asked, are there any plans on how to sync with the BIDS starter kit on the PET front? And yes, we're having examples there, and what we've been discussing also within the BIDS maintainers group yesterday was that it seems to be, especially with those new extensions like the BEP 001 being merged, that we need to be very, very specific about our common principles again. It needs to be very clear what we mean by session run and so on. The wording needs to be checked again. And maybe in the starter kit, there should be something as simple as a comic on describing what do we exactly mean by a run by a session. And this is one of the first things that we'll do now for the PET extension, because there it is really it can be a very complicated setup. So we want to be as simple as possible to make this exceptionally clear what we mean by the different things, so it's not a hurdle for people to convert their data. So that's our aim. So you guys should just let us know if there are any things that you're missing in the standard, and look at our data, or in general, if there are questions about PET, we are very, very happy to answer them or to help you, guide you towards how can we utilize this PET data, what tools are there. because we work with quite a big group of PET developers as well. So please let us know. And until now, are there any other questions? We see here, until now, only Remi, and there's not really a lot going on the chat yet. I fear people are eating lunch. You know, here it's lunchtime.
It is. That is a very good point.
Maybe I can take up a discussion point that I would like to make, and especially that is for PET. That I mean, again, we have to focus a lot also on going back in time and getting like data that's stored on local hard drives at different PET centers and so on. So we have to focus our time a lot on converters, and I'd be very curious to take a discussion on how to best approach this because, you know, if you are a PET researcher and you have data at your local center, I mean, how much does it require for you to take that data and get it into the right format? Do you need to have a software person that can take care of it? Can you do it yourself? And I would like to maybe take a general discussion on some outreach and how we can best convince people that, or help people to convert their data. And for that, I mean, if anyone have experience or would like to, you know, make some points on this, please join in. And at least the way that I see it is that, and that's also something that, yeah, exactly, as also with workshops, of course, as being suggested here. Thank you. And I think that's also the way that we're gonna try to approach this, is that we're gonna have some workshops in at the same time as maybe one of the big conferences that we have. And then let people join in, and maybe they can bring some of their data, and then we can like make a small hackathon or something like that, where we can help people get their data into BIDS format. And then they can take that tool with them back to their respective centers and then try to convert their data. It's just, I mean, it could potentially be a lot of work. So it's just, we need to make sure that we are helping the different centers as much as we can with doing this. And yeah, I think especially what Christophe is saying here, and maybe we could invite him up here if he wants to like elaborate. But I totally agree that I think for most of these centers, it would be a burden. I mean, again, imagine all these different sources of data, and then all of a sudden you're required to get it into a different format that you already have. And then you have to talk to your blood guy, you have to talk to your physicist, and you have to talk to all these different people. And it could potentially be a huge burden to get it done. But that's also why maybe I want to say that a solution as Remi is working on and also presenting here is really great, because it helps you that once you are acquiring the data at the local site, you have to do this while like at the same day or right after you have carried out your experiment, because then you're doing it along the way, and you make sure that once you have acquired your data, you can quickly and easily get it into BIDS format. But it's going to be a potential problem with the data that goes back in time.
Yeah, so maybe then we can get you take the question that's above the character limit. So looking at this discussion of that, when we write the standard now, we hope, of course, to have a more better theoretical understanding of the experiment. One of the things that I don't think is a little bit different between MRI and especially functional MRI and PET is that in terms of tools, we are much more limited right now. There are still, for example, pre-processing has been still, I would say, almost as variable as in MRI, and we have looked at this, actually the influence of pre-processing and also the outcome of statistical experiments after you have varied the pre-processing, and how much that can vary. If I motion correct my data or not has a huge influence. If I do partial volume correction or not has a huge influence. But I have to say, maybe in terms of the experimental setup in PET, there is so much collaboration between different areas, the radiochemistry, then the physics in the scanner, and then the acquisition going on that is, I would say, on the data acquisition side in MR, maybe a little bit hidden. I mean, in BEP-01, it comes out now, but it's more hidden for the person who gets the fMRI scan out, whereas for us, it's very present. That I think in terms of having the theoretical overview of the experiment, you need to have that in order to do PET modeling. So I'm not sure it will help us. Sorry, this was Vittorio, yes, but maybe it is, and I'd really like to see Christophe exactly comment on this, since he is the leader of one of the other large PET sites in Europe.
I'm not the leader, just the collaborator with the BEP-01, and I'm also in a center where we do have on-site PET and MRI. So my opinion is PET requires already, as you said, lots of collaboration, but I feel it's more like a vertical collaboration from the physicist. You need to have the psychotron, the chemists, the radio pharmaceutics, then the injection, then you get the image. While for MRI, this level of collaboration is pretty much the MR physicist and the end user. So you only have like 2 layers, while for PET you have 4 or 5 layers at least, unless you just somehow buy your radio tracer from a company and you get delivered at 8 a.m. and that's it. But that's more of a clinical setup in a hospital. And in a way, because you collaborate vertically, you have less collaboration transversely, because the PET physicists have their own little world. So it's more of a lasagna. And in the end, just getting your images, you've already stumbled on quite a few difficulties. And then a consortium or an agreement, and in a way you ask what would help spread BIDS PET. I get, well, collaboration. And how do you get collaboration? Because you have BIDS PET. It's the chicken and egg situation. So here it would probably be do as best as we can for BIDS PET, and try to present it and explain and re-explain at any meeting and conference with PET. Typically also in MRI, you can have a degree in neuroscience, so more background in psychology or biology or neuro something. So there is a broader range of people using MRI for their own research. PET is typically driven by some doctor and neurologist behind. And we know that sometimes the medical doctors are a bit more difficult to convince. They are trained not to change easily and not follow trends. So if you convince the doctors that organizing their stuff in a way that is that will help collaboration and bring in more money, more grants, more data, then I hope this would break. I guess, I mean, for the radiochemists, for the cyclotron operators, doesn't pay off to organize the end results. So it's really the boss of the lab who has to be convinced that the data, and it takes time. I mean, culture of data sharing. How long has BIDS been around? How long have we been talking about data sharing? And in my own lab, that I've been advocating for that for 5 or 6 years, and we still haven't made any single data set public. I haven't given up, but it takes years to change the mentality of a lab and a field. So, yep, that's my point of view.
Yeah, and here maybe we can explain why we have been working. We have been so active in this, and this is maybe one of the lucky things. So what I just posted is the PubMed link to our SIMPI database. Our head of lab, Gitte Mos Knudsen, who is she's a medical doctor. She's actually the head of ECNP right now. And so she has a complete medical background, but is so aware of all these modeling things. Already in 2005, she ended up establishing a database for PET data. So all of this is available. For example, all our PET data is available. Right now, we have our own archiving system. So we have hundreds of PET scans that we can share, but it comes right now with all the caveats. We will, in some cases, we can give you raw data. In most of the cases, we give you derived data like binding potentials. Also, right now, we're constrained due to GDPR and sharing it with anyone outside Europe, even if we have a direct data agreement. So we're also working very much on that to get it, to develop models so we can share. We're working on establishing an open neuro archive for PET data where we can share this more widely because this data is so rare and expensive. So I think the carrot, like Christophe says, should be on the clinical side because it is the doctors, and you need to, give them incentives to do this. And like Vindu also comment that one thing is making data collection and kind of processing easier. And there we can say, okay, if we make tools like Remi just presented, where you can, as you type, instead of writing the injected dose on a piece of paper, because you measured it from the vial after you've drawn it, you could actually write it directly in this form, and it goes automatically into your JSON that you then send along with your BIDS data set. That would be one thing, but then also especially showing the clinicians the importance of, for example, the influence of their processing, because often each lab has their own established tools and they run this, but what if they just run it differently? What would we end up? Would we see the same result? Would we not? Also, are they even aware of the choices they're making and why? And there we need to make our tools, especially for PET, so intuitive and so instructive, because it's not technical people that work with this. Most of our software is MATLAB, still all of it. We have, at least in our lab, we still have a bunch of code that is I wouldn't say legacy code, but that has been around for a while, because the actual modeling, the solving of differential equations, that really hasn't changed in years. So there's a service to the community that's necessary to showcase, okay, data sharing can help you because you get more data, and actually, yes, we have done the NARPS paper 2 years ago, but nobody notices it because we did it in PET, and we did it not with 70 groups, but we just did it ourselves, running it through 400 different reasonable pipelines. And this shows that once you do this analysis, you see the influence of processing on the results. And we need an input, and we need to, from the clinicians, in saying, okay, we realize this is an issue and we want to help and change this.
One thing also, if I may add, is PET is a clinical tool, and it's very much used in hospital for clinical purpose. While I know that MRI has a clinical purpose in a hospital, but apart for delineating the tumor or lesion in MS, most of what we do in MRI research for neuroimaging, it's not really, and that's one of the weaknesses of MRI, is when is the next clinical breakthrough of functional MRI or diffusion weighted MRI and so on. So it's more the MRI, advanced neuroimaging with MRI that we're doing that tries to move into find a clinical application, while here PET has always had a clinical application. And actually, in the lab where I work, pretty much half of the development of PET radio tracer, new ligands and stuff like that have been sponsored by the pharmaceutical industry, and that's an extra. So when you do something clinical, if it works, you don't change it unless you have a very good reason to change things. Or then we end up with a pipeline that is for clinical and a pipeline that is for research. And two options makes it a bit more complicated. And then pharmaceutical industry, they may not be so keen on data sharing. So that would also be something to keep in mind. And then at the moment you negotiate with the pharmaceutical company that this is the way to go. So if the pharmaceutical company enforces the data sharing or says this is how data should be organized, then we would already have half of the development with this enforced goal of standardizing how the data are represented.
I think that's a very good point. Thank you, Christophe. And I think especially also, as you're mentioning, from the pharmaceutical company. So I can say that, for example, for our lab, we're doing a lot of studies for depression and also now with psilocybin, so magic mushrooms. Again, I mean, often you want to replicate things. I mean, research results, and also if you want to go into a clinical setting, results are always more powerful if you can replicate. So replication is one thing, and also, of course, getting larger sample sizes. So if we can have a study here and we have a drug from a given company and we test it and we see that it has this effect, then it would be worthwhile, or at least I'm sure that I could convince a different group to carry out the same thing, simply to make sure that these results can be replicated. And then I think that, I mean, the point that Melanie, she made before, that we should convince clinicians or other PET researchers to test a lot of pipelines. I'm not sure that's necessarily a good point, because you're also we also just have to remember that that they're potentially opening up themselves to critique, because what if they get a different result if they used a different pipeline? So at least from my point of view, I would try to convince others that they should share data, first of all for replication, but also to increase sample sizes. And I think the PET community in general, they're going to run into these problems no matter what, because if you want to look for a biomarker and do prediction, then if you have only 20 subjects to do this, that's not going to do it. So we need to convince other researchers that they should share the data, so we can make these larger analyses. And if we convince people to do that and they can get their name on a nice paper, I think, at least that's from my point of view, that would be a good strategy. But again, as you say, it takes time to change the mind of all these people. But yeah.
Yeah, and maybe and maybe one of the big points is also that, especially when the pharmacological industry funds PET studies or funds tracer development, of course the primary study will be looked at for the primary outcomes. But especially when we have clinical studies where we include healthy controls, I see no reason why the pharmacological industry could not share the healthy controls for further analysis. That should really not be an issue, but we just need to convince them. And you know, also from an ethical point of view, PET is very costly, like Martin pointed out, and as I already wrote in the chat, it's also more invasive than MR. I mean, we have a limit, for example, on 3 PET scans for a subject. That's it. And this means also that from the ethics side, if we look at this from an ethical perspective, if we do PET scans on healthy control subjects, we should share them, because there's no need to put more people through this procedure. And this should be an argument that should also convince the, I would say, the clinicians and the head of the research lab that unfortunately need to spend money to build this infrastructure and to help their labs share data and put it online, but that in the long run, they will save money, because they will be able to then get healthy control data instead of reacquiring it. And that should be our kind of motivation for getting people to turn to the dark side and share a cookie. Yeah, exactly. People before profits. I also think this is really something that we should simply consider. We're less than 10 minutes now, so we just want to wrap up and ask if there are any other questions. I've tried to copy most of the links in the Mattermost channel, so please have a look there regarding the specification, regarding the different publications we've shared in between. If you run into stuff, if you want to share PET data, if you want to get help analyzing PET data, you know, have a look at the PET-BIDS community and just ping us. Right now, we're still on a Google Doc, but you can even just drop a comment and we will see it. And we are in the process of moving over to GitHub and to Markdown. And once this is there, we'll make a final round of comments for the PET-BIDS spec before we make a pull request. So please let us know if you have input now, because we'd rather have it now than when we're in the stages of the final pull request. That's it from us over here. Thank you so much for tuning in, and we'd really like to hear from you. Sorry, the Google. So what? Hang on, is the Google Doc in chat or Mattermost? So the Google, the Google Doc is basically the BIDS specification. So if you go to this, which is the current BIDS extension, this will actually redirect you to Google Doc. And later on, once we have merged to Markdown, this will redirect you to GitHub. So you only need to remember this link. Cool. So drop us a comment there, have a look, and we'll try to get through this. Yeah. Thank you so much, everyone.
Yeah, thank you so much.